Assalamualaikum warahmatullahi wabarakatuh, and a very good day to everyone. My name is Nabila binti Yahya, and with my friends Audrey Ashrina Elhin and Nur Fatinisha binti Basir. Today we would like to present our findings about solid state NMR, and the title is Solid State NMR of the Yersinia pestis, outer membrane protein ill in lipid bilayer nanodiscs sedimented by ultracentrifugation.
So these are list of contents for this presentation.
Ill is a virulence factor for Yersinia pestis that promotes bacterial cell attachment to human cells and gives the bacteria resistance to innate immune response in humans. We investigated the feasibility of sedimenting nanodiscs containing the outer membrane of protein ill for solid-state NMR experiments. Precipitation using polyethylene glycol or lyophilization or precipitation were previously employed to pack samples in the constrained volume of a mass rotor. High-resolution NMR spectroscopy requires a high detergent concentration, which is incompatible with ligand-binding activity of ill. NMR mapping investigations means tooling, structure, and activity are no longer possible. Protein contained in a lipid bilayer nanodisc is completely functional and produce good resolution. The same samples can be used for concurrent activity and NMR experiments. There are two objectives for the solid-state NMR experiments. The first one is to sediment phospholipid nanodiscs be constituted with ill for solid-state NMR experiments. The second one is to obtain correlated structural and activity information of ill nanodiscs for solid-state NMR experiments.
Okay, for methodology, expression and purification of membrane scaffold protein. The membrane scaffold protein, where is MSP, required for nanodisc formation, will produce and purify in two forms, which is MSP1D1H5, which lack helix 5 of MSP1D1, and MSP1ET31, where has 3 additional helix repeat of helix 4, 5, and 6 in comparison in MSP1D1 sequence. Okay, next is expression and purification of ills. Ills and C-terminal ill type, as the previously stated, is that ills was cloned in E. coli plasmid. Before residues glue one of the native sequence, the express nano acid sequence of the ills is at an addition and terminal melanin. Next is preparation of ills in miscellaneous physical and nanodisc and function of assays. Ills was refilled in N-dicyl phospholipid, whereas we call is DEPC, and sample were made into phospholipid liposomes, phospholipid nanodiscs, and DEPC micelles for solution and NMR. Both NMR spectroscopy, as shown in the text, and the shift in SDS page appearance molecule weight that corresponded to the change from the unfolded to the folded state of the transmembrane barrier were used to measure the protein folding. The H1 NMR peak for the trimethyl ammonia protein at 3.15 ppm was monitored to determine the DEPC concentration. Okay, next is a nanodisc sedimentation at the ambient temperature and 900 rpm. Nanodisc sedimentation tests will carry out using the Beckman average by measuring the UV absorbance at the 280 nm of the supernatant 170 diffraction and both diffraction obtained the 240 centrifuge tube, and the degree of the sedimentation was determined. Okay, next, for sample of NMR spectroscopy. For solution NMR experiment, the result of oil in resource content for 400 milliliter of 0.5 mmPs. The for the sample of pills in MSP was 1D1H5 nanodiscs for solution NMR experiment content 450 liter of the NMR buffer and 0.5 mm. In solution of a volume of 66 liter inside the MAS record, the sample of pills in the sediment nanodiscs for the solid state NMR research content 5.5 mmPs. The sample of pills in the proteolipomase of the solid state NMR study containing 2.4 mmPs, which is 1.5 mg and 740 DMC. Last but not least is NMR experiments. Solid state NMR experiment will carry out using either a buffer 500 MHz spectrometer of it. Utilize a Hans anchor plus sequence with continuous wave 100 decoupling 1-dimensional 31-bit spectra rate 10 on the 500 and 700 MHz spectrometer 1-dimensional 30 carbon and 15 N spectra were recorded using the BC, which contain the period of the 500S or 2NS for H, 1H13C or 1H15N and TPPM sequence for the 1H decoupling. The magnetization transfer time used to obtain in 50N spectra was 2.5 ms, with a transmission time of 1.67 ms and refocusing delay of the 1.11 ms. Next, for proton derived spin diffusion was used to the dimension solid state NMR 13 carbon recollection spectra on the 70 MHz spectrometer with one hydrogen and 30 carbon contact duration for the 40S mixing time of the 40 ms and SWF ppm H1H decoupling.
Now we move to the next part, which is result and discussion. Firstly, we focus on the preparation and solution NMR of L nanodiscs. Why pestis L branch of detergent and lipid sample appropriate for solution and solid state NMR experiment, which is it can recall L. Larger membrane protein and their complexes have been studied biochemically using the longer scaffold protein, which is a MST1E3D1. This preparation has a homogeneity resembling that of anti-nanodiscs and a narrow elution profile, according to analysis by occlusion chromatography. A well-dispersed 1 hydrogen over 50 nitrogen has been correlation. Additionally, homogeneous preparation of L in membrane protein nanodiscs were achieved with some molar ratio. These nanodiscs elute substantially faster than their smaller membrane protein, which is its already counterpart. It's consistent with their greater size and at an elution volume that is comparable to that of anti-membrane protein nanodiscs, according to size occlusion chromatography. These nanodiscs' greater size make it impossible to detect protein signal in the 13 carbon labeled L solution NMR spectra. Only modest signal from protein aromatic side chain location can be seen in the 13 carbon spectrum. Figure 1 showed that it was a nanodisc characterized. Next, we focus on the second, which is the ligand binding activity of L in nanodisc and effect of detergent. The capacity of L to bind fibronectin-coated surface was then examined using ELISS. Both smaller membrane protein nanodiscs and the larger membrane protein nanodiscs can be used to recreate L's fibronectin binding activity. In both instances, L display concentration-dependent binding to fibronectin-coated plates. The 1 hydrogen and 15 nitrogen correlation spectrum of L in 170 DEPC has a narrow. DEPC added to L in nanodisc had a significant impact on the NMR spectra, which is with a peak from the water-exposed lobes being more significantly impacted. The disruption of fibronectin binding in nitrogen molecules appears to be brought on by the interaction of monomeric detergent molecule with important water-exposed site on L and possible also in fibronectin. Then, L and its high molecular weight human ligands such as fibronectin are likely to be inappropriate for chemical shape mapping experiment using nanodisc sample. This picture below shows that L's ability to bind fibronectin in nanodisc and detergent. Alright, move to the third focus, which is solid state NMR of sedimented of L nanodisc. The solution NMR 1 hydrogen over 15 nitrogen spectra obtained after dilution of the sedimented fraction to its initial concentration of 0.5 is nearly identical to the spectrum obtained before centrifugation. This observation, together with the sample apparent transparency following sedimented, lead us to believe that the membrane protein of L nanodisc concentration is not the result of random precipitation. L maintains its structure in the sedimented of L membrane protein nanodisc as seen by the two-dimensional 13 carbon correlation spectrum. The spectrum shows multiple result peaks from 13 carbon. With multiple peaks at the same or similar place, the spectral resolution is equivalent to that observed with L. The open shape of nanodisc may provide better sample concentration and filling than spherical liposomes. This figure shows that the NMR spectra of L membrane protein nanodisc following redilution and sedimentation. The last part in the result and discussion is sedimentation properties of nanodisc. The molecular weight and stoichiometry of protein and lipid nanodisc component, as well as measurement of hydrodynamic of MsP1D1H5 and MsP1E3D1 nanodisc. L reduced the total specific volume and the anticipated sedimentation time in MsP1D1H5 and MsP1E3D1 nanodisc. The ligand that bind the protein contained on the nanodisc, like fibronectin in the case of L, the sedimentation efficiency would be further improved.
In conclusion, our findings demonstrate that centrifugation may sediment nanodiscs containing the outer membrane protein L and transfer them to a rotor for solid-state NMR experiments. For solution NMR analysis, the nanodisc samples can be diluted back to their original concentration, showing that sedimentation has not damaged the protein or lipid bilayer structures. Additionally, since the protein is still active in the nanodisc, the same samples can be used for activity assays, solution NMR, and solid-state NMR experiments. This makes it easier to investigate the structure and activity properties across a variety of dynamic timescales without the need to lyophilize the sample or add potentially harmful detergents or precipitants.
So these are lists of reference that we use to complete this assignment, solid-state NMR experiments. That's all from us. Thank you for listening.